Right, so the last class we talked about monosaccharides and the general formula or general formulae for the triose, the pentose, and the hexose sugars. Uh, right, the biological processes in which they are involved. And we also look at the structures of the pentose and the hexose, the ribose and the glucose molecule. And as for the glucose molecules, we also we saw that it's isomers. So it has two isomers, alpha and beta glucose, right? The ones that we are supposed to know for the syllabus. Now today we will talk about disaccharides and polysaccharides. Right, so disaccharides, as the name suggests, are two molecules of sugar. Can you hear me?
Yes, sir.
Okay. Are two molecules of sugar that have bonded together. Right, so the di, as we are familiar with, is two, and the saccharides is sugar, sugar molecule. So disaccharides are formed, right, when two monosaccharide molecules, right, bond together. Now, what happens is, right, when they bond, they form what is called a glycosidic link, glycosidic bond. And the process is a condensation reaction, something that we have come across earlier. So a bond is being formed, right? And so a molecule, water, is given off. An example is sucrose. So sucrose is a disaccharide, and it is formed when you have a molecule of alpha glucose. Right, so this is where the isomers come in handy. Right, bonds with a molecule of fructose, and that gives you sucrose. And the type of bonding that we need to know. Right, so remember that these are all numbered. Right, so this is carbon number 1 over here, and here we have carbon number 6. So 1 and 6. And when the molecules pretty much form a straight chain, right, this is not as relevant for the disaccharides, as we will see this happening more with the polysaccharides, which is, we just get to, but still, right? So the kind of bond that we have in a straight chain of carbohydrates is a 1,4 glycosidic bond. What this means is when you've got two molecules of monosaccharides, and right, carbon number 1 and the functional groups it has attached is reacting with another molecule at the position carbon number 4, right, with its functional groups, and these two form a link. The other type, right, which is mostly seen in branch chains, and again, right, the straight and branch chain thing is something we'll see in polysaccharides. But right, the type of bond that we will encounter is a 1,6 glycosidic bond. And that, of course, is right, a carbon 1 attaching, forming a bond with carbon 6 up here. Right now, I'm not concentrating on the fact that this is a beta glucose, just right. I'm looking at this as a monosaccharide. So just to show the positions, right? Carbon 1 forming a bond with carbon 6 up here. And that basically happens when you've got branch chains coming off. So what does this look like? Well, right, if you have two molecules of alpha glucose, right, so let's just draw that. You have oxygen, and another one. And the functional groups, so you have H and OH, right, and these are both alpha glucose, right? So this is carbon number 1, and you can see you've got the OH group below. This is carbon number 1 as well for the other molecule. Right. And what's happening is that the two OH groups react. Let me write this so it makes more sense. And this is where the bond is forming. So you've got the entire OH group from one molecule, and just the H, the hydrogen from the OH group of another molecule, and this is carbon number 4, by the way. Okay, and these two are forming a bond, and what you're getting, you're losing a molecule of water, right? So this is a condensation reaction. Condensation reaction basically, when bonds are formed, but it entails a loss of a water molecule. And with that, right, what you end up with, you draw the same thing again with the two molecules. Part. And now you've got right, and the numbering of the carbons. So just to highlight that, this is carbon 1, this is carbon 4, and this particular bond that is formed now is the glycosidic bond. Easy?
I guess.
I mean, any confusion?
No.
Right, so we're not going to details of, oh, what if it were, like we mentioned, sucrose, right? So, oh, what if it were alpha glucose and fructose? How would that look like? What if it were alpha glucose and beta glucose, which also happens, obviously. There's galactose as well. So, right, we're not going into that. Also, right, these are two molecules of alpha glucose, as I mentioned. And the product that you've gotten is maltose. That is a disaccharide, right? And as you know, maltose is what leads up to the formation of starch, right? or glycogen, depending on what organism you're talking about. So this is what a molecule of maltose looks like. Uh, right, all again, you can see I've not bothered with any of the other functional groups at this point because I just wanted to highlight the glycosidic bond and where it's coming from. Otherwise, you've got all the H and OH groups, right? With each carbon, and then the CH2 OH up here with carbon number 6. Right, so if I draw that in detail, just to give you a full molecule, so to speak. So you've got CH2OH, right? That is what it looks like. You put that in brackets. Oops. Yeah, okay. Does that seem nicer to you?
Yeah.
Okay. So, you still, I believe, haven't gotten the hang of the looks of the molecule, but you will. Okay,
Yeah.
so that is about disaccharides as far as the molecule and the bonding is concerned. Right? So what you basically need to know, right? You have 1,4 glycosidic bonds, 1,6 glycosidic bonds, right? This is a 1,4 glycosidic bond, right? Carbon number 1 and carbon number 4. This is maltose. That is what a glycosidic bond looks like. Basically, two monosaccharides joined together by an oxygen, right? And with that, right? So that's as far as we need to know as far as the disaccharides are concerned. But what we're going to talk about now is a certain property that disaccharides and monosaccharides have. Well, most of them, right? And because of that property, they are called reducing sugars. Now, we've talked about oxidation and reduction, right? So, oxidation is either the gain of oxygen or the loss of electrons or an increase in the oxidation number or a loss of hydrogen, right? And thereby, the opposite, reduction, is either a loss of oxygen, a gain of electrons, a decrease in oxidation number, or gaining hydrogen. Right, so these guys are reducing sugars, which means, right, they are reducing agents, which means they cause reduction of whatever other chemical or compound they're with, right, and are themselves oxidized. So that's what reducing agents do. Right, so most monosaccharides, or in fact all monosaccharides, and most disaccharides oops, most disaccharides are reducing sugars. Except, so this is the one that you really need to know, sucrose. So our friend sucrose, right? Table sugar, white sugar, whatever you want to call it, right? It is non-reducing. So it does not act as a reducing agent. And there is a test, a experiment, a procedure that is carried out for this, right, to check this whether a certain sample is a reducing sugar or not, and right, that is called the Benedicts test. Right, so obviously Benedict is a person's name, right, and because it's named after a person, right, what we use is a Benedict's reagent, Benedict's solution, right. Now the detail of the test that I'll be going over, I'll take that from the Cambridge book, because it is much better explained over there. Okay. So what happens in the Benedict's test, right? The Benedict solution is, right, a copper 2 sulfate solution. So copper 2 sulfate solution, right, in alkaline. So copper 2 sulfate is a salt, right? It's an alkaline solution in an alkaline solution. That's what the Benedict's reagent is. And it is blue in color. So as a summary, right, it is blue in color. What happens is when you add something that is a reducing sugar, so either monosaccharide or disaccharide, except for sucrose, right, what happens is the copper, so Cu is the symbol, copper 2 is reduced to copper 1, right, and you see a color change. What you see is that you start getting an insoluble brick red copper oxide. Copper 1 oxide. Okay. So that is where, when you know that the sample that you had, that you used, was a reducing sugar, now you won't be able to identify which sugar it was specifically, right? So this is more of a qualitative test. A qualitative test is one where you can only tell the presence or absence of something, for example, based on certain right, physical properties or changes, right, but it does not give you a quantity as to what you were using, or how much of something changed. So you can't put a number to it. Whereas a quantitative test, so though you've got other types of tests that are quantitative, right? And as the name suggests, quantity is where, right, you have a measure or a value, right, of a product that is formed, perhaps. So how much of the reactants you used and how much, right, of a product you got, or some value, light intensity, temperature change, right, gas release, some sort of quantity, some sort of value. If you're running an experiment on photosynthesis, for example, you measure the volume of oxygen released, or the change in temperature if you're measuring a reaction as an exothermic reaction, or an endothermic, so temperature will drop, right? So if you're getting a value or a measure of something, or a product or a change for a product or a change, then that is quantitative. If you're just looking at changes in properties in general, like you got a precipitate or the color change or something, you can't really put a value to it, right? Then those are qualitative tests.
Okay.
And then there's something called a semi-quantitative test. So it's basically a qualitative test. But what you do is, for example, in this case, you get an insoluble brick red precipitate, right? So what you do is you see, well, how much of it was formed, or how fast, right, or how dark the color was. So these aren't exact values, but depending on how much or how fast or how sorry, not how dark the values, how dark the color. How dark the color was, this can give you an idea of the amount or quantity of the reducing sugar. Now, we haven't got into the procedure of how we carry it out. Just do that, but an idea of the amount or quantity of reducing sugar that you may have had. Again, you're not getting a specific quantity, and that's why it's semi-quantitative. So it gives you an idea, just an idea, right? If there's a lot of precipitate forming, of the precipitates start forming very quickly, or if the color change happened very quickly, if the color change is very dark, it goes to really dark, right? Or whatever other qualitative test you're carrying out, and you notice, right, these things, then it becomes semi-quantitative. It gives you just an idea of how much or how strong or how concentrated, or those kind of estimates. Now, any questions?
No, sir.
Okay, now how do we actually do this thing? So first you need to you take the Benedict's reagent, right, and you need to add it to the solution that you want to test. All right, so here's the procedure. Add Benedict's solution. Let's use reagent to avoid confusion. To test solution. So this is what you want to test, whether or not this is a reducing sugar. Again, you won't be able to tell which reducing sugar it was, right? But you'll only be able to, okay, fine, maybe it was a monosaccharide, maybe it was a disaccharide. And you, right, put it in a water bath to heat it up. Right, you want to heat it so that you get energy for the reaction to take place. If a reducing sugar is present, what you will notice is a color change, right? It will go from blue to green to yellow to orange and then red brown. So remember, the Benedict solution was initially blue, and it will go to green, then yellow, orange, and then red brown precipitate.
Yeah.
Now, again, the detail in which I'm going is not given in the Edexcel book. I'm taking this from the Cambridge book. You can
Yeah.
stick to you can stick to the level of detail that you have in the Edexcel book, but this is just so that, right, we have a better understanding of what's going on in this particular experiment.
Sure.
Right, and one thing to do is to use an excess of the Benedict's reagent. Why are we using an excess? So we are able to react all of the reducing sugar that we have, right? And so the more the reducing sugar that we'll have, the more intensity of the red color, right? So the concentration of sugar, reducing sugar, will be proportional to the intensity of color. And as I already mentioned, this is a semi-quantitative test. Now, in case, right, this is what will happen if you've got a monosaccharide or a disaccharide that is a reducing sugar. In case, right, you have, so let's make this 1 and let's make this 2, right? If you did not have any change happening in the first test, right? So initially you just add the Benedict solution, the Benedict's reagent to your test solution, and you hope that the color change happens when you heat it, right? If there is no color change, that means that what the sample that you had was a non-reducing sugar. Is non-reducing, right? Which means it's either not a monosaccharide or maybe it's a disaccharide like sucrose. So what you do is, right, you need to hydrolyze it. So what you do is you add hydrochloric acid. And what this will do is it will cause the hydrolysis of glycosidic bonds. All right, so just going back, these bonds that are formed to make a disaccharide or even further polysaccharide, right? If, for example, you had a sample of liquid starch or some other polysaccharide in an aqueous or liquid solution, right? And so the hydrochloric acid will do is break it up into its monomers. So yes, you get glucose or fructose or galactose, and we know monosaccharides are right reducing. Achha. Once you've hydrolyzed it, then we'll carry out the test again. And hopefully, this time we'll get a red brown or a brick red precipitate. right? Which means whatever we had was either sucrose or a polysaccharide has been broken down into its monomer units. If in case there is still no change, right, that means the test was a negative. Right, and whatever you had in this case was not it was not perhaps even a carbohydrate, right? If that happens in the second scenario, if that happens in the first scenario, right, sample was not a reducing sugar. So that we talked about, right? In that case, you go to method number 2. But if in method number 2 you still don't have any change happening, right? Then that means that the sample probably wasn't a carbohydrate to begin with, right? Because we're talking about reducing sugar. So even if by adding hydrochloric acid, it was hydrolyzed and perhaps you got amino acids or fatty acids and glycerol, right? They're not going to give you a positive result for the Benedict solution test. Is that, I mean, were you following along? Did that make sense?
Yes, sir, it does.
Okay, that's good to hear. And yeah, so right now we have some time. And so I move on to the next chapter. That was the end of chapter 1A2.
Yeah.
And so now start chapter 1A3, in which we will talk about polysaccharides.
Okay.
Right now, poly means many, and saccharides means, right, sugars. So you've got many monomers, and by many we're talking about, well, anything above 3 basically. But as we'll see, we've got like hundreds or perhaps even thousands. So anything 3 or above is a poly as far as we are concerned. Okay, and they're all joined together by glycosidic bonds. Now, a certain technical detail, right? Even among the polysaccharides, if the number of sugar units are monomer units, right? If monomers that are forming the polysaccharide are between 3 and 10, then, right, they have a special name. They are called oligosaccharides. Just something to know, right? And right, if they have 11 or more, then those are called true polysaccharides. All right, we already talked about the fact that, right, glycosidic bonds are formed by condensation. And so you have loss of a water molecule for each bond formed. Keep that in mind. Not in total, for each bond formed. Right? And right, these bonds are broken. By hydrolysis, as we saw in the Benedicts test. Right now, in a biological system, they're going to be happening by the action of enzymes. Right? In as far as sugars are concerned, right, in alkaline conditions, right, so in the mouth or in the intestine, right. So chemically, it can be done by hydrochloric acid, right, uh, in a laboratory experiment. In your body, it's happening in alkaline conditions mostly, right. So they can be broken by hydrolysis, which is basically, right, under those conditions with the enzyme and everything, a water molecule is added back, which makes sense, right? Because you want, if you're breaking it up into its monomer units, you want to recover the OH groups for each of the monomers that were involved in forming the glycosidic bond. So you need to add back that water molecule. And again, this is for each bond. Okay. Uh, then, right, we're going to talk about what polysaccharides are good for. So one thing we know is, right, their biological uses. You obviously have energy, right? So glycolysis in respiration for the production of ATP. You have these guys as an energy store. So you have starch in plants, right? Glycogen, humans and mammals. Then you also have cellulose, right, which is structured, right, so it has a structural role. In plant cells, right, you've got other sorts of polysaccharides that are involved in bacterial cell walls and fungal cell walls, right, so chitin or murine, right, so those are other polysaccharides. So you've got chitin in right, fungi, and murine in bacteria. So the cell walls are made of those polysaccharides. And yeah, you even got, right, what we'll see later is that they act as, right, part of proteins, so part of protein molecules, not exactly protein molecules. So in the cell surface membrane, you've got proteins that act as receptors or other functions, and right, protein molecules or lipid molecules. And so in those cases, molecules and they have various functions, biological functions in different situations, right? So it's not just for energy. It is a primary role that these guys have, but not the only one. Any questions?
No, sir.
And right, I don't think I can squeeze that in, so I will mention it again in the next class tomorrow. But polysaccharides are not as reactive and they are not as soluble, right? So that's why they make up a good energy scope. So I will go over that. Okay, so that is all for this class. And we will continue this in the next. See you then.
Yes, sir.